now. Okay, so welcome everybody. This is the first lecture, or I would say a seminar of a series of seminars that we trying to build here at the International Hellenic University. As you know, every academic institution has a PhD program. So here we have such a program. Well, every PhD program needs to aim high. Otherwise, why invest 3-5 years in doing it? And we think here that successful or well-rounded and all-around programs should also have a coaching initiative. Something that will help the PhD students persevere the burden and the difficulties of executing their PhD, and also to help them establish a vision and see what's the point of all this. So this seminar series aims to provide career advice from top experts in the broader field of academia. You may see here people from data science, or biomedical fields, or from molecular ecology. So it doesn't matter. We share here the experiences, and which is a common ground for everybody. And as said again, we try to give you not only PhD students, but also postdocs. We always need that. Even us, PIs, need that. And master students, inspiration, a career vision. Why not give you contact to top level professionals, expand your network, and make new interactions. I will close it here by saying that what's the next slide? Yeah, you always need to dream big. 80% of success is just showing up, and success is incremental gains every day. So your success will not be in one moment. It will be a daily procedure that will build up to the day that you defend your thesis, you present your master topic, or you succeed in something. With that said, I would like to welcome today Maria Georgiadou. I already mentioned in the email that you have received that, well, she's successful. However, I know from firsthand that she did an excellent PhD thesis. Cell and cell reports, etc., papers testify for that. And wherever she got EMBO fellowships, she got senior.
It's okay, I'm going to talk about those things.
Okay, so you're gonna brag about yourself. So without further ado, the floor is yours, Maria. Thank you.
Hello, thanks.
You would like to share your screen, I suppose.
So yes, good morning, everybody. I hope you can hear me well. And I'm trying to check who is on online now, so that kind of have a view of the people. There are no pictures, so I cannot see your faces. It would have been nicer to have it face to face, but I am in Ioannina, so that's not possible. I put together a couple of slides, I have to admit, from older presentations, which I have given while I was in Finland. No, share screen, yes. So it will look a little bit like bragging about myself, exactly what Spiro said as a joke. It's not about that. It's just that I thought I would tell some stories. It's like a lot of text here, to tell some stories, and you can interrupt me anytime. So it's basically my career path with some examples there that I thought could be of interest that could bring some discussion. So I'm from Greece. I'm from Thessaloniki, and I studied biology in Thessaloniki at the Aristotle University. And that was, I got my degree already in 2004. And I could say something already from the start, that I was in the beginning hurrying about everything. As a student, even as in school, I was always in a hurry. In a hurry to get language diplomas, in a hurry to finish the school, to finish university, to do everything in a hurry. This is important for the future. That's why I mention it here. And then while I was in biology, I realized I liked more this molecular biology, something related to human disease. But in Thessaloniki, there is not that much about that. So then in the department of their genetics, the professor that was the most known was Professor Konstantinos Triadafilidis, the father of Alexandros Triadafilidis, that many of you know. So I applied for him to do my thesis, not based on the topic, but as said, based on the fact that he was a good group leader. So then my thesis was about genetic identification of the Greek partridges, alectores. These are birds, basically. And all I did was some DNA extractions and PCR to identify nucleotide RFLPs, nucleotide polymorphisms. And there I was also in a hurry. I had to do my task, everything that was for the whole year. I wanted to do it the first 3 months. So then I got some more job afterwards. And then while I was in Professor Triadafilidis' lab doing my thesis, he came to me and he told me, well, I'm applying for funding for a PhD position. And if I get it, would you like to start PhD in my lab? So that, wow, great. That sounds great. Yes. And up until then, I think he had never missed a grant. He was getting all the money. I mean, everywhere he was applying, he was getting the funding. So it was kind of set in my mind that, yes, he will get the funding and I will start. And it was, again, some genetic identification about sheep, if I remember correctly, or I don't remember exactly which animal, but it was again with animals. And genetics, basic genetics. So I was quite happy and excited, but then when I finished already like half a year later, he had not had yet received funding, the results from the funding. So he said, well, I know this professor from dental school who has a project and it's related to what you have done already. So would you like to work with her for a while until you start the PhD? I thought, why not? So I started working with this professor in the dental school. And but she had no idea about genetics, so she was basically a dentist. So I was going in her office at the dentist office, taking some blood samples from the people going there, and then doing in some labs in biology, again, DNA extraction and so on. So I was also, she had a grant for that, so I was also dealing with the financials. I was basically doing everything for that, but I was not paid as a biologist. I was paid as an unqualified technician or something like this was my position. So I go to the bank once because, you know, they from the university at that time, you were getting paid with I don't remember how it's called in English, but anyway, that you get this paper saying you get that amount of money from the bank. So I was going to the bank to get the money, and I say, oh, I came to get this a great amount of 380 euros. And she was super pissed at me, like, how dare you? How old are you? How dare you saying so that you are even paid something at your age? I was 21. And you consider this amount so low and you are rude and this kind of, you know, I was shocked. So then a couple of months later, the professor to the Philippines calls me in the lab and tells me, unfortunately, I didn't get the funding, so there is no PhD in my lab. And I was like, oh my God, what am I going to do now? That was what I had in mind. So, but then he calls me back and he tells me where there is a lab in pharmacy that got the funding but has nobody to work on the project. You can go and talk to the professor. Okay, there in pharmacy, it was a lab working with pharmaceutical plants, nothing to do with what I was doing until then. So, okay, why not? I go there, have an interview with the professor. Then I had interviews with other professors there. It was quite complicated whole procedure. So eventually I got the position, not started as a PhD student. I started half a year later as a PhD student because all the processes were quite slow. And as said, here is the title that the plan was to isolate and characterize these metabolites, the terpenes are called, and the terpene synthesis, so enzymes from a plant called Salvia fruticosa and another similar one, Salvia pomifera. So that was the project. I worked 1 year in the lab, and well, my salary was 660 euros per month, and I was really lucky to have funding because most other students doing a PhD, there we were quite many in the same floor doing PhD or master thesis and having fun together. So most of the others were doing the PhD for free. So this was my story from Greece. Well, and it continues in the next slide to say how I actually decided to, what triggered me to move. But is there anybody who wants to ask something related to my Greek experience in the past? If not, then I continue. Okay, sorry, you don't see, I didn't put it in, now I have this here and I don't know how to put it in.
Full screen, it's fine, we can read it.
Sorry about that. I apparently am not that good with
Do you see,
Zoom.
you want it full screen? Do you see next to 92% a button at the bottom of your screen?
92%? I don't see 92%.
Do you see the clock of your computer, the clock?
Ah, yes, here.
On the left, there is 92%. Left. Yes. Well, next to the clock, a little bit high. Left.
I don't see that.
It's okay. We can see the slides.
Anyway, well, sorry about that.
No worries.
I don't see 92%. So I don't know what to mean. But anyway, so as said, I had started the PhD. And while I was doing this PhD in pharmacy, then while walking basically the stairs of the building, I saw this poster, this biology at work. This was a poster from EMBL. EMBL is a European Molecular Biology Laboratory, has the headquarters in Heidelberg. At the moment, it also has institutes at many other countries in Europe. And they have this very prestigious and very competitive doctoral program. I think, I don't know nowadays, but at that time they were getting 50 PhD students per year for the whole EMBL labs and all EMBL institutes in Europe, and they had thousands of applications for these 50 students. I didn't even know about EMBL at that time, but then I saw this poster, and it was a PhD student symposium from EMBL. It was basically a symposium organized by the PhD students from EMBL for PhD students around Europe. And I thought, oh, that sounds cool. So I applied, I got a fellowship to go. So I went there, and I thought this was absolutely amazing. Well, I didn't understand much about the topics discussed, I have to say, other than, these career paths and all this. But about biology, I didn't understand very much because my knowledge wasn't that advanced. But then as I visited there, EMBL, I talked to other PhD students that have been doing PhDs in different countries. I was really in awe, like, wow, that's where I want to go. So I went back to continue the PhD in Greece, but this this thought remained in my heart and mind the whole time, that I compared all the time, like, oh, how it is here in Greece and how it was there at EMBL and how many things they were learning and this and that. Even I liked even their coffee breaks and the coffee room, everything. So anyway, I wasn't also, even though I was getting along really well with the people in the lab and the whole department, I wasn't getting along with the professor very much. So after we had kind of an argument, I told him, well, I quit. And that was in September 2006, so about a year after I had started in the lab. So he thought I was just joking, but I wasn't. I said I'm quitting, and the next day I just didn't go back. So then himself and other professors from the department, they were calling me. And they were kind of threatening me also. They were telling me, if you quit once, then you will always quit. You will never succeed. This is going to follow your entire career. So they were trying to keep me there with this in this way. Well, I didn't have any help how to find a PhD abroad, but I was certain that that's what I want to do. So I started applying for at different places while I was working at my parents' shop. And then, as I say here, I'm happy I didn't keep these applications. That's because they were quite funny, as I didn't know how to write the applications. And of course, I applied for the MBL PhD program, but as I said, it was super competitive and I didn't get in. But I was really desperately wanting to go there. So I was on the floor crying, saying, that's where I want to go. I don't want anything else. And eventually I got an email from another doctoral program in Belgium saying that, well, we found your details from the EMBL, and would you like to apply to us as well? So I applied, went for interviews, and eventually got a job offer and started there my PhD. So the interviews were in March, and immediately in 2 weeks they gave the offers and agreed that I would start, but then I was starting the next October. So basically from September to October, I wasn't working in a lab. And so then I went to Belgium. Why doesn't this change? Yes. So I went to Belgium. I went to the lab of Professor Peter Karmelit in October 2007, as I say there. Peter Karmelit is one of the biggest labs in the world. It was so big that there were only the same lab with the same PI, no subgroups. We were at that time something like 15 PhD students, 15 postdocs, and 20 technicians. So it was enormous. And then there were many other smaller groups that we were all sharing the same huge lab. So it was a great difference It was intimidating. But also when I went there, I thought, okay, it's going to be hard. I got that. People were telling me also when I went for interviews, students were telling me, because we were allowed to talk to the people in the lab, we had a tour in the labs and so on. They were telling me that he's super difficult, he's not nice. And I could even tell it from the interview. He was seeming the best person. He was smiling, encouraging, but you could tell he was hard. So, but he was so encouraging that even though I went there to the interview telling him that, I don't have a master's. I didn't mention that, but maybe you saw it, that I have only done my biology degree. I went directly to PhD without a master's. So I said, I don't have a master's. I don't even know if I'm eligible here. So in front of me, he picks up the phone, calls the secretary to ask if they can deal with this problem, and tell them that if there is, to ask him if there is any technical problem, if not, that he's fine with that. So they told me from the secretary, we will tell you in 1 week, and then they told me the same evening. So he was quite efficient with these things. But well, the other positive thing there is, by starting in a PhD doctoral program, then you start with other PhD students at the same time. So I didn't go there completely alone. I had already, when I was going to Belgium, when it was the first time leaving my country, first time leaving my home, first time speaking English, first time living alone, all kinds of first times. And I was quite stressed about that. Then the good part was that I had already met a couple of people that were also starting in Belgium at the same time, and we had someone there to know. And that was very, very good. And these people are still my friends. So anyway, I start in the lab. I didn't even put here the name of the project. The project changed every single year. Every single year, at the end of the year, he was telling me that this project is not going to lead to a cell paper, so we stop it. And when I mean we stop it, means that I have been working day and night. I didn't go holidays. I didn't go. I didn't have weekends. I was working day and night, and all the data were in vain because he was not interested in those projects. So every year I was on the floor crying, saying, I have to quit, but I will not quit. So it was quite tough. Another thing that I was thinking to tell you, yes, something else that I wanted to mention is that when I started, maybe you remember my previous projects that were with animal genetics and plants. So I was going there to work in angiogenesis. So with human cells or mouse cells, because or in mouse experiments. So I had absolutely no clue. I didn't know what is a cell line. I didn't know how to do cell cultures. I didn't understand basically anything. We had to do histology, molecular biology. I had not seen them. We didn't have even these courses in the university. So he was, even though he was notoriously difficult with the people, I think he saw that I was very motivated and enthusiastic. That's why he offered me the position. But then he also allowed me to spend some months to learn all the techniques. So about half a year, I was just following people and playing in the lab, because I tried everything, but all the techniques to learn everything with fake samples, or not only fake, some that I destroyed and they were not fake. So Leuven was a great place to be, because it's in the center of Europe. And well, apart from the fact that you can travel everywhere, and that was also super nice, it was also a scientific hub. So we had speakers coming to give talks from all over the world. leading scientists. And of course, because it was Peter Carmel, it was known, he was inviting all these leading scientists, and then we were going out for dinner with them. So I had met Nobel Prize winners or other people that won the Nobel Prize afterwards, and I had lunch and dinner with them and drinks with them. So that was really nice to see them. These are real people, normal people, and to see that, yes, I could be like this. It's not something that they're not aliens or something. That's one. And then, of course, you feel more comfortable when you go to a conference. Then I went to talk to in a conference, and many of these, the organizers of the conference knew me. So I didn't feel, I felt quite welcome. It was easy for me to interact, to network there, even though I went to the conference alone and it was in the US. Then what else I could say? Well, do you have to ask anything related? Because I not all them.
I was apart from your tell us from this experience. Clearly, this experience changed you. Like
Yes,
you came from let's say in football terms like League C to Champions League immediately. It's like
yes.
taking playing from a local team to a global scene. Okay, one thing from each: the worst, if you can say that, and the best experience in this period, if you can tell them.
I'll try to remember some things. Of course, the worst was that indeed Peter was, as I said, notoriously difficult. So when I wrote that, I think it's this one of the memories I have. For there, the PhD programs are a little bit different in different universities. So there, you have to write a pre-thesis, a smaller text to apply to your advisory committee. And then if they give the okay, then you can start writing the full thesis. So when I wrote this first draft and sent it to Peter, I got an email back with big letters in the title saying I gave up. So telling me this is so bad, so horribly written that I gave up. That was his point. So he was talking like this. He was threatening us. Like he was sending emails. At that time we didn't have phones, right? It was before 2010 when the cell phones came, and then nobody had even in 2010. I got mine in 2013. So you had to be behind your computer the whole time. So I got emails at midnight telling me I need this by 8 in the morning. And he was super furious if you wouldn't have it. What I did, of course, because it was impossible to have it by 8 in the morning, what I did was to immediately answer that you will have it ASAP. And that was 10 in the morning. So we were working like crazy and under a lot of pressure, so much pressure that the last 2 years of my PhD, I got psychosomatic problems. I was going to the doctors, couldn't find what I had. It was really terribly difficult. So that was the worst part. The best part was, of course, that I could do really amazing science. I still remember some moments where some big discoveries that we were like, wow, look what works. It's amazing. We had a hypothesis and now we did this experiment and it's exactly like the hypothesis. And this is like really amazing. And it's so wonderful when you can just do anything, because there is a lot of money and you can do anything you want. When we were going and saying, okay, which antibodies should we buy? He said, buy them all. His attitude was like, shoot, shoot, shoot. So you take all and you try them all. So you shoot, shoot, shoot. That's what he was telling. So that was more work, but anyway, you could do anything you wanted. As said already, one of the best things was meeting all these people and having dinners with them, having also dinners with my friends out every evening. So we had a very good company. Many people from many countries around same age. So we had a very nice group, and all these people are now professors somewhere in Europe with a lot of funding and different topics. And it's really nice to have this kind of network. What else? Yes.
Just to conclude, again, continuing with a comparison with the football terms. So you experienced, if I may say so, bullying, both in Greece and also in Belgium.
Yes,
so you experienced like the difficulties, like to wake up early, to go out in the weekends, etc., etc. But the difference is why you didn't quit in Belgium and you quit in Greece is because of the trophy, a different trophy you were about to.
It wasn't though certain. So about 50% of the people in Peter's lab were quitting. So I think that was the dropout rate. About half of them were leaving. And then also the PhD students were so burned out that I think maybe one or two of all the PhD students that were at the same time as me eventually continued to academic career. Everybody left academia, which is not per se bad. I mean, there is many other options which I had a list to show you later, so hopefully we have time for that. Yes, what because you had put there about the papers, and you were already asking me at some point to mention about how you get a cell paper. I can tell you a couple of examples there. So when I first went to the lab, I was put next to another PhD student who was doing the proofs of his paper, his first author paper. He was one year in the lab, and he had a first author paper, equally contributing with a postdoc. Like me, it was the same for me. My papers are equally contributing with a postdoc. So he has his first author paper. It was a cell paper and in angiogenesis. And for that paper, Peter Karmelit got an email from Judah Folkman to congratulate him for the paper. Please write down the name Judah Folkman and check it out. Judah Folkman, he passed away that year, unfortunately, but he was the person that envisioned angiogenesis as therapy, inhibiting angiogenesis as an anti-cancer therapy. And so he was the pioneer of the field, and there are many anti-angiogenic agents given to patients since actually the times that I was in Peter's lab. So not only he was having a cell paper, he was having a cell paper that was so important in the field. So yes, and I was like, a cell paper? I have never read a cell paper. I didn't know that people can publish cell papers. So that was my experience the first month. And then, as I said, I started in October. And then for a Christmas present, from Peter, I got a Nature paper for revision. So he gave me a paper that was submitted to Nature to revise it. I had never read a Nature paper, and I didn't understand a thing from the Nature paper because, as I told you, I didn't have any experience before. So I started reading the names of cell lines for people that have worked with cell lines. They are called like B16, HCT, 116, and this kind of thing. It's like, what is this Chinese here? So, of course, the only corrections I could do was spelling mistakes. But then we were a team of people, PhD students and postdocs, that we were supposed to do this. So after whole Christmas break, being super stressed about this revision, we met with these other PhD students and postdocs, had a discussion, and then I started learning how to do that properly through the discussion with other peers. So more senior peers, obviously. And so by doing that, I had reviewed many Nature and Cell papers. So by the time I had to write my own, so then you learn what you need. You learn what you need to ask. You learn what the others will ask because you have asked them yourself. Well, of course, you need to read this kind of papers to know how they should be. But it wasn't something out of reach. If you were in Peter's lab, it wasn't out of reach. And this was the biggest difference. Because when we submitted to Cell, actually we had submitted to Cell and Nature with 2 weeks difference. Then eventually it was Cell and Cell Metabolism, the 2 papers. But when we submitted to Cell and Nature first, we had a super big party there in the lab with beers. I don't think it's allowed anymore. So we were celebrating the submissions. We didn't care about the acceptance. Submission was already super hard. So then we had a 1-year revision. The questions we were asked were super difficult, but then you need the PI and yourself need to decide that, yes, I may do these revisions. Or you can say, no, it's too much for me. It's too much money, too much time, too much effort. I cannot do it. But in order to go from, of course, I'm still talking about cell metabolism versus cell paper. It wouldn't go to very low paper, but still to go from cell metabolism to cell, it's a big difference. So there you really need to invest much, much more. We made new mice and genetic engineered mice during this year. We actually rewrote the paper. We made many new studies, but we were also working in teams. I wasn't working there alone. One person alone to get a Cell paper or Nature paper is only by luck. To get it really through hard work, you still need to be more people. One person is, I would think, nearly impossible. Okay, I go then move to the next. So I then moved to Finland.
Maria, sorry to interrupt. There is a raised hand by Vasilis.
Yes.
Yes, Vasilis.
Yes. What about like during your PhD studies? Like if you come up with new ideas, can you maybe work on them afterwards, or are you like limited by the funding and the orientation of the professor?
I had a lot of we had a lot of funding, so that wasn't a problem. If you are in a lab where there is not much funding, then that is a problem, of course. As said, I was changing project every year, and the project we were until nearly the moment that the project was looking really like it will become a Cell paper. He didn't care. So we were doing anything we liked. So basically everything was our own idea. Maybe he was giving a hint in the beginning where we should go, but then everything we did was our own ideas. We were discussing, we had frequent discussions with him to show him data, and he was giving some directions. But as said, we were so many people, he couldn't give directions to everybody. So yes, we were free to do things, which was sometimes not good. But of course it was fun. Yes, but then we couldn't, then at the end when he was seeing that, okay, this story looks really nice, this can go high, then you had to do what he was telling you to do. So we had freedom, I had freedom for 3 years, let's say. And then the last 2, I had to do what he wanted us to do, because it was so much anyway, I couldn't do anything else. Yeah, was this your question, or did you have more to ask related to that?
So like, because there are some like structured PhDs, these are maybe like worse than the regular with the.
Oh, you mean like those that you have a funding and that is with a specific topic and you have to do these specific, give the specific deliverables and so on?
Yes.
I have not worked with this in Greece, so I don't know how strict it is in Greece really, but I have had my own funding as a postdoc, this kind of funding in Finland, but also other people in Belgium that I could see. So if you publish something, then usually the funding agencies are happy. So then you have to say that you have to explain why you have to diverge a little bit or a lot, but you kind of show it that it is a little bit, because usually the people that review those are not experts in the field. So you say that I diverted a little bit because of this and this reason. And then I actually did these other things instead and I published. So here it is. So I don't think I ever followed what I wrote I will do. So
Okay. Yes.
is this clear?
Yes. Thank you.
Yes. Yes. So then I moved to Finland for postdoc. I went to Finland because Dr. Pavakostas was in Finland already. So I had to find a postdoc there. It wasn't my plan initially. And not only in Finland, but actually in Turku, a quite small place. So I couldn't continue exactly what I was doing before because there wasn't such topics. And again, I selected the best lab. So I asked Spear's group leader to give me some names of people that are that are known to be good in their work and as people. He gave me a list of names, and then I sent an email to Johanna Ivaska. She's here in the picture, and I'm the one there in the corner. So I sent her an email, and she didn't reply. And I thought there was no open call, but I just sent her an email with my interest to work in her lab. Then I wait about a month and I send a friendly reminder. I was quite shy to do that, but I thought, well, who cares? I'll do that. And then she replied immediately and she invited me. I had a Skype interview and then she invited me in the lab. And it wasn't the best time of the year. It was dark and it had a lot of snow. Well, it was beautiful with the snow, but well, I was complaining in Belgium for the weather. Imagine in Finland. And then, okay, we agreed that I will start and that I would start in September. So in the summer, I started applying for European postdoc funding. And then I was awarded the EMBO fellowship and I went to Johanna's lab. And this was really a great lab. There, it was the complete opposite from Peter. She was super understanding, super nice as a person, supportive. She had also a good big lab with an international lab and publishing very well. So it was very good lab, but not bullying, as Spiros mentioned before. So again, I said here it's the topic. Before I was studying cell metabolism and angiogenesis, then here I worked with basic cell biology. The topic was how integrin activity was regulated by metabolism, but I basically didn't do any metabolism. In the meantime, I had children, so I was also out of the lab for a while. And then after a couple of years in Johanna's lab, I started thinking that I would like to do something more translational. And I had that in my mind. So at some point, then I got another fellowship for 3 years. And we thought that, okay, now as we are going to stay another 3 years, I say here we bought a house. I say here to show that we thought, okay, yes, we are staying for the 3 years of my fellowship. But in the meantime, by chance, so a friend told me on a hike, so it was really just a chat. Oh, there is an opening at Orion. Orion is the biggest pharma in Finland, and they also have basic research. They have R&D, research and development department, and basic research. And they said, oh, they have an opening for senior scientist in oncology. I said, what is this? Let's check it out. That was Thursday that we had that chat. And then I thought of it while I was working in the lab on Monday. And then I said, oh, let's check out the, what is this position? And I check it while doing an experiment in the lab. I check it on my phone. So when is the deadline? Today. Today? Okay, well, let's apply. Why do I have to lose? So in the evening, after the work, after the kids, I started the application. And because I was stressed, I was writing the application only on the online form. So the website crashed and I lost everything. So I had to start again and the deadline passed. So I actually applied after midnight and I thought, okay, now it's not even, I'm not even within the deadline. But anyway, they invited me for interview, but we were apparently 2 people that went to the final interviews, but the other person was more qualified because they don't tell you anything about what you are going to do in the company. It's all very confidential and secret, but apparently at that time they were working with prostate cancer. So the other candidate was working on prostate cancer and I had no experience exactly with cancer. So, so they got the other person, but 2 weeks later they call me and they tell me, okay, we liked you as well. So we opened the position for you too. So I went to the company like this. And I said I started working in oncology department in a team that all of them were working in prostate cancer. All of them, all the others but me. So then that wasn't too bad in a sense because I got to be the lead of a new project that was not prostate cancer. Nobody else had to take it. So, and that was nice because it was a preclinical project that I started from the beginning. And then until the moment I left Orion last August, so August 2022, I was leading this project through all the phases of drug discovery and preclinical phase. And now they are, they're waiting to start clinical trials. So I even was involved in planning the clinical trials, but just the plans. So it's quite late, so I'm not going to tell you much about what I did at Orion, even though
Maria, excuse me.
no,
Because it's too late, I think people would like to interest, and I know you're talking excellent about this, the differences between academic mindset and private company mindset. That would like something. That would be something interesting to hear.
Okay. Well, maybe I could say that in the company, it really depends on what kind of job you do. So I was doing, again, basic research. And so I was again in the lab, but there the different mindset, I had also a lot of money, like with the other positions. So there it wasn't a difference. So sometimes in the company you have a lot of money compared to university where you don't. But for me, that wasn't the case. Then another difference in the company is that sometimes they have specific projects like the one I said that I had to join from the beginning. And that's what you have to do. You cannot have your own ideas. But again, I was in a position where I had to find new ideas, and those were only coming from me. So I could do whatever I wanted there as well. That was half of my time was dedicated to that. In the company, you have a lot of meetings, constantly meetings. which is quite annoying. But on the other hand, you get to discuss with people from many different fields and see many different views, ways that you would never experience otherwise. So I don't know, for me, the company experience was amazing. I was coming, as I write here, I was coming out of my comfort zone every single day because I said I had to talk with chemists, and I had to try to understand what they were telling me, or I had to try to explain to them my point of view in a way that he would get it. Or I had to present my ideas to the upper management, and I had to bring there also not the science only, but also the marketplace, like why would this be a good asset for the company? I had to discuss with business people, and for everything you do, you need to. It's not always the same. And so I learned a lot of things, a lot of new things. As said, also the projects were very different. So also related to science, I worked in many different topics. So yeah, for me, learning is the driving force. So that's why I liked it so much. Well, maybe there the most difficult is that they might stop your project at any moment without good reason for you. That's because it comes from the management, and you might not understand the reason. And it's quite frustrating because you have spent so much effort, energy, you feel this is your own project, and then it's stopped. But again, I had that experience from before, from my PhD, and for me, it was not too awkward. At the company, you work with teams. It's always a teamwork. It's never, there is never like one person show. Or some people try it, but it doesn't work well. So that's again something important. I think I have a couple of slides here that might be related. So here it was some advice that I wanted to give to you when you're looking for your dream job. And the first is to find your passion, indeed. And when I'm talking about find your passion, it's not exactly I want to work with plants, or I want to work with animals, or I want to work in angiogenesis or cancer. As you see, you can change topics and you can still be passionate about it. But it's more like, do I want to do basic research, or I want to work with computers, and I like working on the bench, or I don't like to work on the bench. I want to work like something more business oriented or not, this kind of things. So this is what you need to understand: what interests you more, what energizes you more, and then which are your values at work. It's important. Like I want to really I do it because I want to build the future. I want to train people, or I just do it as a job. For me, it's only salary. It's fine. Like family is more important. I want an 8 to 4 job, and I don't want to have this overtime. So there also you need to think which are your skills and strengths, and again, not just which techniques I know, but I am more introvert person, or I'm more team player. So these kind of things, personality also, not just technical skills. Of course, technical skills are important. Well, I have that personality part later. If you like to work alone, or you want to work in a team, and then this is how often do you want to change projects? And if you can handle this in the company, you will have to change projects a lot. That's for sure. Yes, and the expectations is related to what I have said before. So, but also related to your ambition. So is it okay for you to go to a job and just stay in that position forever, or you are very ambitious and you want to have a clear career path, like it is in academia, for instance, that that is you always improve, like can rise in position. But in the company, you might just stay senior scientist for 20 years. Most people in the company were senior scientists for 20 years. It wasn't boring. They were doing different projects. They were learning new things. They were senior scientists for 20 years. So it's quite hard to go higher in a company. Yes, and another advice is to explore your options. Talk to people. Try to find information, of course, from internet, LinkedIn, but talk to people. And yeah, when you make decisions, try to consider your priorities related to what, for instance, I said before. Yes, and
Thank you.
Yeah.
Thank you, Maria. Can't thank you enough for sharing your experiences. You shared your time with us, with so many students from different backgrounds and levels. And yeah, I think your advice and your experience is so precious. You saw us working in a low-level lab, going like to something bigger, changing countries, and even combining the private industry with academia. It's difficult to draw general rules, but I hope you all got some valuable input. I think what Maria said, like find your passion. That helps you, both yourself and the principal investigator, on the one that will hire you. We want to talk with people that have this, they are crystal clear on what they want to do and if they are good fit for the job. And chase your dreams. I mean, and work as a team. Is it right? I mean, a common trend that I saw in your presentation is always the best work of your life were done in a team environment.
Yes.
Not as an individual behind an isolated space, not talking to anybody. So, like both in Belgium, in Finland, any company, there was team effort that made the most out of it.
Yes, everywhere. I worked everywhere as a team. The first thing I did when I joined my postdoc lab, Johanna Ivaske, was to tell her, I have learned to work as a team. In her lab, the people at that time were not working as teams. Everybody had their own project. So I told her I want to work with someone as a team. I want to share experiences, emotions, results with someone daily. I want to have a PhD student. And yes, I got a PhD student, and then there was another postdoc that joined a couple of months later who was sharing same feelings. So we three worked very close together. And if you look at my postdoc publications, we are all three almost in all publications because we helped each other.
Thank you. Timothy Jenkins, our next speaker, is online. I don't want to.
I would greet you.
Yes.
Thank you so much.
Thank you very much.
Have a good rest of the day.
Thank you. Thank you. Timothy, I mean, I'm so grateful by sometimes the people that are accepting to share their experiences. I mean, Timothy is like another high-level principal investigator. He did his PhD at the university, if I read correctly, your CV, at the University of Cambridge, right? I mean, how often do you have the opportunity to come in touch with people that have such an experience? I did my PhD at the University of Greece. Not that it's inferior, but I mean, wow. And you accepted to talk to us. So I can't thank you enough. And you finished your thesis at 2019. And by now, like how many? 4 years later, you are associate professor. So that assistant professor, like super quick, you're on a fast track. And I know from Thomas Dacios, our contact liaison, that you have such great experience and so many super things to tell us. We can't wait to hear from you. Unfortunately, some of the participants are working at like a private sector, so they asked me if possible, and it's okay to you to record this lecture so they can follow it later. Is it okay with you?
Of course, not a problem. And thanks, by the way, Spiros, for the invitation. It's always a pleasure to interact with you guys, and I'm more than happy to be there. Although now you set the bar very high for me to meet, so I hope I can.
So Mas told me that is it that you and your PI are paid by the hour at the level of 5,000 euros per hour? Is that right?
I haven't done the math, but
yeah. Like so again, I stop speaking and we try to enjoy you. You have the floor. Thank you.
Thank you very much.
Let me give you the permission to share your screen.
Should already work, I think.
Okay, it's already there. Yeah, thank you.
Perfect. So I was asked today to talk about two primary things, also due to the interest scope, both of your university, but also the participants. The title itself is more of a catchy one. It might have changed to the one that you saw originally, but I just preferred it. So I'm going to talk a bit about some pig work that we've done, but also snakes, since that is one of the central aspects of our research. But my expertise really lies within computational biology and applying computational tools to therapeutic discovery. So very briefly, I already got a nice intro. I did my undergrad in Australia at James Cook University. As you can see, it was a horrible place to spend some time. The slogan of the region was "where the rainforest meets the reef," and I have to say it lives up to those expectations. And I worked a lot on venomous animals there, did zoology and ecology actually originally, but got more and more interested in other aspects of biology, specific human health, and then went on to Cambridge, where I did my PhD in human microbiome. So I did a lot of next generation sequencing, looking at how the microbiome changes in the context of autoimmune conditions such as multiple sclerosis. But I still had this fascination with venomous animals and antibodies. So that is where I then joined Andreas Lausen, the professor and head of center that I'm at right now, the Center for Antibody Technologies at the Technical University of Denmark. And in case you're a bit unsure on where exactly that is, we're located right next to Copenhagen. And that is that nice little green speck in the bottom. The big green part is also officially part of Denmark. And that's Greenland that Trump also wanted to buy not too long ago. But that's not where we are. We're down in a very nice city. You can see in the background one of the most famous streets in Copenhagen. And right now, this is actually representative. We have good weather, which is not always the case, or I might even say not often the case here. But right now, it's a very pleasant place to be. And in the bottom, you can see my research group. I've got a few master students, bachelor students, PhD students, postdocs. Not all of them in the picture there, but we had a nice snowy outing in the winter as well and had a snowball fight outside. So with no further ado, I will talk a bit about the research that I'm doing. I'm interested in both fundamental questions of how antibodies interact, specifically with a lot of different targets. So as you're probably aware, with COVID, we had all of these different mutations of the COVID strains. My idea is more focused on snakebite, but there we have a similar problem of a lot of toxins that are quite similar. And we'd like to use a single antibody for as many of these different variants as possible. So I want to understand what really dictates these interactions of cross-reactivity. But I'm at a technical university. I'm not just interested in understanding, I also want to do something about it. So my research lab, the Digital Biotechnology Lab, is also interested in translating the science. So how do we actually make therapeutic solutions to some of these problems that we've identified? How do we make solutions that are cost effective and are actually translatable to a real setting, and not just a cool idea in the lab, but when you actually try and take it out in the real world, you can't afford them because they're way too expensive, or they just don't work because your proteins are expressing really well in your bacteria. But when we're talking about producing them in scale, you need to do it in yeast or mammalian cultures, and there they don't work. So how can we take these considerations into our workflows early on? And finally, entrepreneurship is kind of the next logical step here, is how do we actually take this out? How are we the people who can guide the process of commercializing these into real life products that can hopefully help people? And so I'm going to talk a bit about all of these aspects, but with a primary focus on the latter two, the translational aspects and the entrepreneurship. And actually, one of the points I really wanted to make about entrepreneurship is a way that we encourage entrepreneurship in my group, but also at the center that I'm working at. And that is via something that we've coined Invention Club. And the reason I wanted to present it is because I think it's something that's quite easily implementable anywhere you're at. It can be within your classes, it can be in your research groups, it can be within your group of friends, really, if you're interested in this and are as nerdy as I am. But I highly recommend that you have a look at this, because the idea here is really to do something that is similar to Journal Club. So you might have heard of Journal Club, where you come together, you find a really cool publication that came out. One of you has to read through it. All of you has to go through the paper, and then you come together and discuss some interesting points around it. You quiz it. That's great in theory and can be really useful. But why don't we twist this concept a bit and apply it to entrepreneurship? Because a lot of scientists are trained to be great academics, but they're not necessarily trained in the thinking that is required for translational research, for actually making these things into commercializable ideas. So the idea here is really that we want to have it like Journal Club, but with your own unpolished ideas that we discuss. So the way I usually describe it is the following, where we have an idea that participants, that can be bachelor students, it can be professors, it can be anything, anybody who wants to, comes to present a scientific concept or idea to the rest of the group. We usually try and run it every second week, more or less. And then they present that idea in a pitch format where they tell us, what is the problem? Why is it currently unmet? What's your idea to solve it? And also, why would people care? Will people actually buy this idea? Is it substantially better than what is out there to an extent that it's worth doing? And then they pitch this in a friendly setting where we ask questions and discuss after. And we facilitate all of this by having some nice alcoholic and non-alcoholic drinks at the end, that we can keep the discussions going and make it a social experience as well. And so this is kind of the general concept. As I just mentioned, you have the pitch, you have a Q&A, drinks and social, and then we usually select whoever wants to pitch next. And I have to say, it's really nice that typically the people who are most active and most interested are the young students, bachelor students, master students. They love this and they do excellently. Here I've got an example of one of my master students, Vlad, who presented a really cool idea on how we can deal with plastic waste using a biosolution. So how can we make enzymes that degrade plastic better and faster, which is really cool. And you can see here, this was, I'd say, medium turnout. We often have quite a few more, but a good mix of students, postdocs, professors. We even had the head of one of the sections there. And it's usually a really nice event after which we can just go outside and enjoy ourselves. And we got a lot of really good feedback from the people who've participated, which is why we're continuing it, and we've upscaled it now to the whole department. And we've actually had some success stories that have come out of it as well. So we've had one startup company that was actually one of the first ideas pitched in this setting. I have to admit, it was before my time, so I can't take credit. But a really cool company that is still going. It's about 5 years old, has 4 full-time employees, has raised over $1 million by now. And it's focusing on developing snakebite diagnostic tools. And that started in this exact setting where we looked at this and he was like, yeah, this could actually work. Let's see how we can get some money for it. Let's try and build this into a company, and it worked. And one that I've also been involved in myself now is this Immunite project, which is more recent. It's about a year and a half old. And we've raised over 3 million kroner, which is, let's say, about 400,000 or 500,000 euros in the last 12 months. And this idea also started with a pitch in this setting. So the idea here really is that you can use your questions, your ideas to start ideating. And one of the key questions I always get in this setting, but I'm a bachelor student, I don't have any great ideas. All of my good ideas are taken already. How do I know what's needed? Well, now you have things like ChatGPT that can give you a bit of a boost, where you can ask ChatGPT, what are the greatest unmet problems right now? Okay, boom, you get a list. Then maybe take a technology that you're personally really interested in, whatever that might be. It can be antibodies, it can be something totally unrelated. And you say, okay, how could I solve this problem with antibodies? And it'll give you some ideas. It's not perfect. I'm glad it's not, because then your ideas would be worthless. But it's very good at starting this process of, you have this block and trying to figure out what to do, but you can just put some keywords in and start building your idea quite quickly now. And you could do it without ChatGPT, but now it's just a lot easier and a lot more accessible. So really no excuses to start playing around with entrepreneurial ideas within the biosciences. Now, overall, at the Center of Antibody Technologies, we've got a lot of different areas that we're interested in. Our primary focus so far has been on snake bites, but we're also interested in bacterial diseases, parasites, industrial biotech and bioagricultural tech. And we use a lot of our experiences with antibodies and antibody-like proteins, binding proteins like nanobodies, to influence these systems. And this here is a list of the companies that have come out either of the center or also personally founded by Andreas Lausen, the head of the center. And you can see it's quite a number now, and they're all alive and successful. I think the connection just dropped, but I will continue. And one of these companies that I want to really focus on today is Back to Life. So Back to Life is our most recent company that was founded, I think, something like 4 years ago. And the focus here is looking at bacterial gastrointestinal infections. And there's a lot of different GI infection problems. You've got things like cholera, you've got agricultural issues, and it's all under this whole context of antimicrobial resistance and the problems that we're seeing with broad spectrum antibiotics. So how can we fix something around this problem? And I'm not commercially involved in this company. I'm academically involved. I collaborate with them. So my opinion here is, I'd say, not unbiased, but I'll keep it as unbiased as I can. I have no commercial stake in this. But the idea with this company was, let's take some of the expertise that exist in our understanding of pathogens, an understanding of infections, and our understanding of antibodies. So we know that there are good bacteria and bad bacteria in guts, and these can sometimes be quite easily identified based on the surface proteins that they have, the way that they're shaped. And if we're able to specifically recognize them, maybe we can figure out what happens. So here in this graph, we've got a classical example of a bacterial infection. The orange ones are pathogens, the blue ones are the good bacteria. And if we give antibiotic treatment, we basically nuke all of them. Everybody gets affected. You will get these antibiotic resistant genes that suddenly sprout up. They'll develop resistances, and now you'll have both the healthy and pathogenic bacteria with traits that make them antibiotically resistant, which is becoming an increasing problem, as I'm sure you're aware. And our idea was to specifically inhibit only the pathogenic bacteria. It's not via an antibiotic. It's just by targeting proteins that are made, so the toxins that are made by the actual bacteria. So here we've got these. How can we neutralize the toxins that they're making? But how can we also prevent these bacteria from adhering to the gastrointestinal lining? So how can we stop them from sticking in the gut and just flush them out as quickly as possible? The idea here was we're using antibodies. So this is just an overview of different antibody formats. But specifically, we're using nanobodies. And the reason why is quite a long explanation, but these VHH binding proteins are small. They're naturally occurring in camel's milk. We know they're safe to ingest, and they're very easy to engineer and work with. So what we did is develop, or Back to Life did, is develop a first product known as Ablacto, which is targeting actual gastrointestinal infections in pig. There are a lot of pigs in Denmark, and a lot of them get diarrhea quite early on in their life. And unfortunately, that diarrhea is not just annoying, it's often lethal for young piglets. So how can we prevent these piglets from dying? How can we improve animal welfare, and also for the farmers, make a product that's cost effective and increases their productivity? So post-running diarrhea has quite substantial impacts. Like, the mortality is not super high in piglets, it's around 4 to 8%, but it creates major weight loss, which then results in a huge production loss, which is up to 1 to 2 billion euros every year. So it's a massive economical problem. And so we've got these binding proteins now that are able to target toxins, that are able to target the fimbriae on the surface of the bacteria. And the idea is that we can mix this into the standard pig's feed to see if we can prevent these toxins from wreaking havoc and creating a lot of problems. And this here is a very busy slide, I'll apologize, but basically what we've got is just a visualization that this binding of these bacteria and linking them together to basically cluster them so they can't really do much. They can't adhere to the surface. Seems to be working. So this is just microscopy pictures. Here you can see they're free and happy. Then we add our binders. They start getting clogged up with binders, and then we've created these films of bacteria stuck together. And we can also see in the lower pictures that this is reflected in their attachment on the actual surface of the gastrointestinal lining, which is great. So they seem to be doing the right thing. We also validated that this is a very specific effect. This doesn't just attack any bacterium there. It's very specific to the pathogenic bacteria that we're targeting. And that's a good sign because we can already say it's a more specific treatment than the broad spectrum antibiotics. But the real true challenge is showing that this also works in a pig setting. So we also did pig studies. We've actually done a few now. I've been involved in one of these. And we saw that when we administer our different antibodies, we are able to save a lot of the pigs from dying, which not a lot of them were, but anyway, that's good. But we massively increase growth. So they actually get a lot fatter, a lot faster, and a lot healthier. It's really cool. So the first paper has already come out. That was the early study. Now we're working right now on a second publication where we've done a bit more work. We've got bigger cohorts. We've actually tested them a bit more thoroughly. And here you can see we've got a 20% increase in body weight of the piglets, which again is a lot if you're talking about each kilo being money to the farmer, and also obviously the resultant welfare of the pig being a lot higher. So this was a quick run through Back to Life as a company, a success story, I'd say so far. It has raised over $5 million and is still running quite well. So there's a lot of cool things. And if you're interested in that space, there's also quite a few job postings regularly. So keep an eye on them. But given that I promised to not just talk about pigs, but also snakes, I will live up to that promise and move on to my snake work right now, and also how we're using computational tools increasingly to try and solve snake bite. So the snakes are pretty much everywhere. They're distributed across most continents around the world, but I have to say that the primary problem space is not in Denmark, shockingly. Probably also not in Greece, although more so. It's primarily in India and sub-Saharan Africa. And it's quite shocking to most people to hear how many people actually are affected. We've got close to 3 million people every year that are envenomed. And we've got close to 140,000 deaths every single year, with an additional 400,000 people that are permanently disabled every year. If we put it into the context of, for instance, the Ebola crisis, which was basically solved within 3 years, this is as much every year as over the course of the whole major outbreak that was then solved. The main reason why snakebite isn't solved and infectious diseases often get solved quicker is that the risk of snakes coming on a plane to Denmark and affecting the rich people there is very unlikely. Infectious diseases travel a lot more into rich countries. So rich countries deal with rich people problem a lot faster than with poor people problems, which is a sad but honest truth. And that's also why we're trying to attack this problem from an academic setting, where we have a luxury of having grant money, public money, that we can use to try and take some of the costs into our hands and develop a product that then is self-sustaining and viable once we've developed it. So that is the idea, that we want to try and tackle snakebite. Now, as I mentioned, why are we doing this in Denmark? It's not a problem in Denmark. Denmark is known for Vikings, average beer. It's okay. I'm half German, so I'm obviously biased here. But they have some pretty good pastry, so I recommend the baked goods. But why snakebite? Well, the reason is there's a lot of know-how in Denmark. They have a lot of antibody companies, a lot of large-scale pharmaceutical companies. There's industry support. A lot of the technology is there, and we're using quite high levels of technology, which you can't always access no matter where you are. So that is why we're doing this in Denmark. Now, the true issue is that right now, the only available treatment for snakebite is traditional antivenom, which is not a therapeutic, actually. It's a blood product, because it's generated in a very old-fashioned way of taking a venom of interest, injecting it into a horse, waiting for up to a year for the horse's immune system to generate antibodies. You take the blood, you purify it, and you inject that into a human. It works, and it's saving lives across this world, but it has a lot of issues. It's very expensive, it's hard to scale up, and it has a lot of adverse effects. And the most important thing really is that it's not very specific. You can't really target it. It's very good against big toxins that are very immunogenic, where the immune system reacts. But it's very bad against small toxins that are not very immunogenic, escape the immune system. And often these toxins are extremely lethal. A lot of them are neurotoxins that shut down your nervous system. So our idea at the Center for Antibody Technologies really is to develop specific monoclonal antibodies that target the most clinically relevant toxins. So we can really have a perfect switch solution where we can target the toxins we need to. We have a pure product in the end, and we know what we're doing. It's a clear, defined therapeutic dose. Now, that is a cool idea, but it's also not an easy one. We have now proven in a few publications that it's possible. We've got some cool antibodies that seem to be working quite well, that are neutralizing whole venoms. So we've got cross-reactive antibodies that can neutralize whole venoms of snakes, which is a great first step. But the problem still remains that snake venoms are incredibly complex. And what I mean by that is that we have a graph here, which is a network plot, where each single node, gray circle, represents a single toxin. Each of these toxins is a toxin we know that has currently been reported, so there are probably some more out there. And the way that they're clustered is based on their sequence similarity. So we can overlay this with some functional information. We can categorize these toxins. So you'll be able to see some of these neurotoxins that I mentioned, the short neurotoxins, the long neurotoxins, but also more enzymatic toxins, metalloproteinases, some of the phospholipases, serum proteases. But I think the overview is that there's a lot of things to deal with. If you're talking about COVID, we know there is COVID spike protein. We need to target this. Maybe there's some escape variants. Okay, so we've maybe got like 6, 7, 10 different targets we want to deal with. Here we're talking about a space of maybe 2,000 different targets. So that's a lot of different toxins to deal with. And if we make 2,000 antibodies, which we can, that's fine, that's good. But the product will be incredibly complex and expensive. So to give you an example, one of the major projects that we've funded right now is to try and develop a solution for sub-Saharan Africa, where we've got over 20 different species of snake. Here you can see some of them. We've got some mambas. This one is a famous snake, the black mamba. We've got some of the spitting cobras and some of the vipers that are pretty nasty. This here is actually one of the world's most lethal snakes. It's the saw-scale viper. And we're trying to make an antivenom that can deal with all of them. So if I highlight this on the plot I just showed you, this is the toxins that we know are from the snakes that we need to deal with. So maybe it's not 2,000 anymore, but we're still talking about like 100-ish toxins. 100 antibodies, problematic. Because, as I previously mentioned, snakebite affects people in impoverished regions around the world. It's not regions that have the socioeconomic power as you have in the US, for instance, or in Northern Europe, or anywhere really in the Northern Hemisphere. It's a tropical disease. And the people that are affected can't afford to pay thousands and thousands of dollars for a treatment. So if we make 100 antibodies and produce them, manufacture them, the treatment cost would be through the roof. And yes, maybe a patient in Denmark could afford it, maybe a patient in the US could afford it, but not the people who really need it. So we've done, as I mentioned, we want to understand the product before we try and develop our solutions. We've done a cost analysis. Now, this is a very rough guesstimate of what we can see are the major factors influencing the final cost of our antibody product. So if we take our idea, take it away from an academic setting, put it into industrial setting, where do we end up? Is this feasible? And so what we have here is a simple graph where on the left, you've got US dollars. So this is costs of goods manufactured. That goes up to this $1,400 mark. And then we've got here the ratios. So how many toxins can be neutralized by a single antibody, 1-to-1 or 1-to-3. Because the more toxins one of our antibodies can take care of, the better, obviously. And also how cross-reactive are our antibodies. And just as a reference, I have the so far best antivenom that was available at the time. It's been out of production for over 5 years now. because it was not cost effective for the pharmaceutical company to actually continue manufacturing it. But this is kind of a golden mark. If we hit this, where we're already in a good place, ideally we want to go under it. And you can see that both the ratio is very important, but also how cross-reactive we can make our products. So the more cross-reactivity we have in our antibodies, so how many different toxins they can deal with, how few antibodies we can use, the better. So to put this into perspective, if we say these toxins here are clustered based on how similar they are, so maybe, just maybe, we can get an antibody that can cover this group of toxins. Maybe we need a couple, but maybe we don't need 20. Here, the same thing. Maybe we can just use one or a few different antibodies. And now we're suddenly in a space of having a product that is commercially viable and can solve the problem. Because then we can also say, these guys, the vipers, they've got certain types of venoms that are more representative with these types of toxins. The cobras are more representative here. So now we're at very low numbers of different antibodies, if we can engineer them to be as cross-reactive and broadly neutralizing as we hope we can. And again, that's where I showed in the papers, we think we can. It's not easy, but it's a solvable problem. But how do we actually make our antibodies cross-reactive? Well, there's a lot of lab-based techniques that I can talk about, but the focus of today should really be our computational tools. I want to excite you, I want to thrill you, I want to scare you, because I have to tell you that where we're at now with computational therapeutic discovery is incredible. A year ago was a different world, and really things have changed dramatically over the last 12 months. And I will try and highlight this throughout the next couple of slides, really. But first, let's take it calm and easy. We're going to start with talking the different ways that you can actually use AI, machine learning, in the process of our antibody discovery. I've got 3 different applications. I stole this from a paper here, but it nicely covers the way that we are currently looking at machine learning tools in the space of antibody discovery. We've got the predictive purpose, where you say, I've got my data set. I want to predict something in this data set. So you don't have a tool, you try and make a tool based on the data you have generated. That's the best because it's the most specialized on your particular problem, but it's also the hardest because you have to generate large amounts of data and you need to be very good at machine learning. The second point is optimize. You have an antibody that is okay, but you want to make it even better. You want it to make it bind stronger. And then the third, and in my opinion, one of the most exciting processes is generating something from nothing. So saying, I want an antibody against this target, let me press a button. And how close we are to this now, I will illustrate. But let's start with the prediction. So one of my PhD students, Max Overath, who is also co-supervised by Simon Olsen at Chalmers University, just across the pond in Sweden, is working on a cool project where we're trying to really address this first point. Right now, there are very few high-quality antibody data sets out there. So we can't just build a machine learning model. We need to generate the data to build this machine learning model, especially for the purpose of seeing if we can predict whether a given sequence of an antibody will be cross-reactive and broadly neutralizing to a series of different targets that we're interested in. So in his project, he's going to be generating data. He's going to take computational data of paratope-epitope interactions. So epitope being where does the antibody bind on the target, paratope, where does the antigen, so the toxin in this case, bind to the antibody. This is very important nomenclature because this is the areas that are most important on the antibody, most important on your target. They are the things that are actually interacting and interfacing. And then use all of these data to generate a machine learning model, most likely based on deep learning, but let's see, the field is moving crazy fast. So things might change by the time we've generated all this data. And then seeing if we can predict whether antibody A, or in this case, here this antibody is predicted to bind not just snake toxin 1, but also snake toxin 2 before we even have to test it, because that will save us a lot of time in terms of solving this whole target space that I've showed to you before. The cool thing is he's already gotten some cool data. So we've done some combination of antibody display technologies. What we have here is quite complicated, pretty looking plots that are from FACS data, so fluorescence-assisted cell sorting, where we take these yeast cells that display the antibody, and on one axis we have expression, on the other axis we have binding. So we want to make sure our antibodies are expressed on the yeast. Then we've got an antigen that is labeled, so our toxins are labeled, that comes in, and if that also glows in a different color, we know that the antibody is expressing, but also binding the target. So in this case here, we've tested against different snake venom species. So you've got all of these different species of snake. You've got the types of toxins we've tested, so metalloproteases, phospholipases. And the cool thing is we actually saw some cross-binding. We're starting to get some cool numbers in terms of how many binding sequences we have, non-binding, because keep in mind, when you're doing machine learning, you don't just want to have your result, you want a gradient. You want to know every antibody that does the job, that does it great, that does it poorly, that doesn't do it, because all of this information helps the algorithm learn what is predictive for a good binder. So don't just take your zeros and your ones, you want the full gradient of data that you can collect. And the really cool thing was that we already saw some pretty substantial cross-reactivity, because if we just look at the sequences of the two phospholipases and align them, we see that they're actually not that similar to each other. There is some significant overlap in terms of sequence identity, but overall it's only 40%. So they're not too similar, which is exciting, because it means we can actually cross-bind quite distantly related toxin proteins. But then we also wanted to see, okay, how about the structural similarity? Because sequence is one thing, but structure is another. And if we actually look at the structure, it's not that different. And the reason I put this in there is if you guys are working with sequencing data sets of any type in the context of proteins, make sure you also assess the structure, because the structure provides a lot of information that you shouldn't neglect. If we looked at the sequence here, I would have told you no chance that this binding can happen. If I look at the structure, I'm not surprised at all. They are very, very similar. You can see one toxin in gold, the other in blue. They're basically on top of each other. So this is how we've started looking at predicting. With generating the data, then we need to build our algorithm. But this is going to take a bit of time. In the optimization, I've also got a series of students who are looking at different ways that we can make our antibodies better affinity binders. So how can we make them bind their targets better, tighter, so they don't let go? And again, there's different computational tools. And in this case, we took one of our recent publications where we've basically in the lab taken an antibody that was okay. We used some lab-based techniques, such as chain shuffling, to improve our antibody, and then we got a good antibody out. So we started with around 90 nanomolar affinity, which is all right. But then we have our final antibody that was 9 nanomolar or so affinity. So a factor of 10 improvement that we saw when we did the lab-based technique. Now, unfortunately, I can't tell you a lab confirmation of what we're seeing, but we wanted to test this in one of the computational tools that we saw to see if we can use something called protein hallucination to suggest some changes in our antibodies and the amino acids that could potentially lead to tighter binding. And the way it does it, this is quite a complex graph, but in a simple way is it looks at the structure of your antibody, it looks at the structure of your target, it does a lot of calculations, it understands how proteins work based on its deep neural network, and it will then be able to say, maybe this amino acids could be better and it will lead to a bit tighter binding interaction. So maybe try and shift that there. And it'll give you a lot of different suggestions that are visualized in these logo plots. So each of these here is an amino acid. This was our starting sequence. This is a particular region on the antibody that we're interested in, the main region that is responsible for binding, namely the CDRH3. And then we went about to actually look at different parts of suggested sequences that we could change. So these here. Okay, not super clear, but then we used some metrics that predict how well does the different sequences that it suggested bind. And now this is our outcome where we say, okay, maybe the final sequence that we want to try is one here and one here. So the larger the letter, the more consistent it is across the different suggestions. So here we've got basically two suggested sequences, 168, 284. So now we're going to go into the lab, try and make these antibodies and see, do we see an improvement in affinity? If we do, yay. If not, okay, fair enough. But at least this is a start of using these tools. It's new. These things are very new, that their work needs to be confirmed in a lot of cases. But it's good to start using these tools and understanding how they work and what their potential is. Now, as I promised, the wow factor is definitely in this space where we start saying, I've got a target, let me generate something. So I've got some other students who are also working on this problem. And what I have to say really changed the field that we're looking at is a concept that came out of the image generation world. So you might have come across the terminology of stable diffusion. It's made the rounds in the context of things like DALL-E 2 or Midjourney, which have been image AI generators where you say, I want a monkey riding a bicycle while in space, and it generates an image for you. And for instance, DALL-E 2 is from the same guys who invented ChatGPT, OpenAI, using some of the same ideas and infrastructure. And this is here just in the background, an example of such an image generator. A more fun example is this one here, which I generated for one of my lectures to illustrate the fun you can have with these technologies. I hope you can recognize these two people. On the left, we've got Marie Curie. On the right, Albert Einstein. And I wanted Marie Curie to serve the ways like a boss, and Albert Einstein to look like a rapper standing in front of his golden Lamborghini. And I have to say, the AI did a pretty good job at representing this. And you might ask me, okay, Tim, why are you wasting my time with this? This is fun, but not useful. Wait, because you can apply the same technology to proteins. Because what they did here was this stable diffusion that really made it so much better. And stable diffusion is based on the concept of introducing something known as Gaussian noise into images. The way we traditionally trained image generators is that you'd say, this here is an image of a cat. This here is another image of a cat, and here's another one. And then they get a lot of images of cats, and they know the absolute truth of, okay, this arrangement of pixels represents something that the humans call cat. Great. But it wasn't so great at making new things. It was good at recalling and identifying, but not making new things. So the way that was attempted to overcome it is for us to help it learn the relationship of each pixel to each other. So they did it by introducing something known as Gaussian noise, where you start randomizing the pixels in the image more and more and more until you get pure noise. And then you take that whole series of data, so the totally noised image, the medium noise, the full image, and say, this is all the cat. And you use that to train. And it started learning the relationship of each pixel to each other, how they're connected. And then you could take Albert Einstein pixel mess and mix it with a Lamborghini pixel mess and start making these cool processes. And you will actually see in a lot of cases, it starts with this pixel mess and starts making an image that's sharper and sharper and sharper. We're slowly putting them together into context. So actually, when it's generating these images, it starts from this and works back into this. And the same thing can be done to proteins. So one of the most famous structural biology groups in the world, the David Baker Lab, is based at the University of Washington. And they did exactly this, where they took protein structures and they took each atom in the protein structure and started noising it, making it more crazy, more crazy, more crazy. And taking this data then to train an algorithm. So we have stable diffusion, but instead of images, on protein structures. They come out with this preprint where they introduced the idea that you can actually now generate proteins, you can dream up these new proteins. And the really cool thing is you can use it for binder design. And when I saw this, I was like, okay, cool. Can I trust this? Up until 12 months ago, as you might remember me mention, the field was very different. If you wanted to make a binder computationally, you would have to make around 100,000 plus different variants. You take them into the lab, you test them, and maybe you get a couple that are good. Buying 100,000 genes is incredibly expensive. It was cost inhibitory. You couldn't really do it unless you were one of the major labs. And honestly, it made no sense. You're way faster to do it in the lab. If you've got an antibody library, you can do phage display, yeast display, way faster, way cheaper if you have that library already. So why would I do this? But what they reported is that they tested for 6 different proteins or so, that they made 100 different binders and had somewhere between 1 and 10% success rate. So 1 to 10 of the things they generated worked. which is crazy because now we only have to buy maybe 100 different variants or 50, which is still not cheap, but it's very much affordable. And when I heard that, I was like, okay, great, this is cool. How does this actually work? So the visualization here kind of demonstrate what we saw also with the image. You start with a mess of pixels that come together, and it starts trying to figure out what assortment of pixels, or in this case, structural atoms, in which conformation do they best create a tight fit to your target protein that you give. So you present the target structure, thanks to AlphaFold, that's very easy. And then it generates a protein to it. And I liked it a lot. I liked it so much that I wanted to see if we can apply to snakebite. So I reached out to one of the lead authors, Joseph Watson here, who put me in touch with a grad student at the Baker Lab, Susana Torres. And we started working on this problem of making AI-generated binders to our targets. And honestly, it shocked me quite a lot as to how fast it went. This is just a rough outline of a pipeline that Susana ran. So this was all to her credit here. But she decided that we should try a couple of different types of designs where we make them 100 amino acids long, 120, 150, and we made 1,000 designs each. So we made a roughly 3,000 designs to try and see if we can bind us to short neurotoxins and long neurotoxins, since these are some of the most lethal targets. And then we can use some filters to try and figure out what's going on. Here you can see an overview of the two main metrics that we use to assess them, PAE and PLDDT. And you can see that we've taken 3,000 as our designs, and we selected 36 based on cutoffs that we chose. We then wanted to test them in vitro, because that's really where you see, are you just making up numbers in your computer, or is this actually meaningful? And long story short, there was some iterations, but we found very high affinity binder to long neurotoxins within the 1 nanomolar range roughly, and also a very good binder to short neurotoxins where we're at the 300 picomolar range, which is insane, since in the lab we've been struggling to get any binders to this toxin for over a year. And this all happened within, I'd say, a month. If we are even faster, this was sending over stuff to the US and back and forth. If you actually wanted to look at the raw pipeline, you can do this within a week, which is pretty incredible, because now you can actually do this for any target you're interested in. You probably don't care too much about snake venom toxins, but maybe you care about some cancer proteins or some parasitic proteins or something else. And this is rather accessible. Like, it's not something that you need to be a coding expert. They've published it in a GitHub repository and even as a Google Colab notebook. So you can actually click through buttons. You don't need to code at all, and you can start designing it. It helps if you know how to code. I'm guessing quite a few of you here will have some experience in coding, so that's a plus, but you don't need it. And it's very accessible. And I started teaching my third-year bachelor students yesterday for the first time. They're getting the cutting edge, because this still isn't officially published. It's a preprint still, but I have seen in my own hands / in collaboration with the guys who developed it, it works. So I just wanted to give you this as a wow factor that you can really start thinking about how you can use these types of tools in your interest, because you don't have to do therapeutics. You can make a lab reagent, you can make a diagnostic, whatever. It's basically playing with proteins. It's protein Lego. And it's starting to be very doable, and you don't need crazy compute for this either. I've run this just on my laptop. Not in that scale if you want to do thousands and thousands of generations, but you can just play around with it on your laptop and start getting some cool results. So yeah, besides that, I want to end on a note that I've talked a lot about hardcore science, and hopefully I haven't totally overloaded you. But I'm very excited about all of these things. I think it's also very important that you enjoy the science that you do and the people you do it with. So as you can see here, we've got a lot of fun activities as well. We've gone to conferences in Italy together with my group. We've gone canoeing, kayaking, and it's very important that you enjoy the environment you work in, you enjoy the science you do, because if you bring these two things together, then you can really do some great work and you won't get burned out. You can work hard, but you can also just enjoy the process of working hard. So on that note, I just want to fund a lot of the people involved here, and I would be happy to answer any questions.
Wow. Can't thank you enough. If I raise the bar so high, you exceed it. So Thank you so much. And we already have a question from Dimitris Dafnoudis. Can you also turn on your camera if it's possible?
Yes, I'm at work, but now I can. Sorry. Hello, can you hear me?
Yes.
Yes. Nice presentation. Thank you very much. It was very informative. I have a question about the snakes toxins. I don't know if, because I'm not an expert in toxins, I have actually never studied toxins in my life. I have a question about if toxins are cooperate in order to give the effect they have. Because
So
if it does, maybe you could benefit from that. I don't know.
So if you mean by cooperate, if they have synergistic effects, then the answer is yes. So some toxins will facilitate each other's toxicity. It's still not fully understood, but there's some toxins that basically make holes in membranes, and those holes let other toxins in and then destroy inside of this. So that is something, if you understand it well, if you take out one of these players, as in the membrane, forming toxin or penetrating toxin, then probably you can also prevent the toxicity of the other toxin. So it's something that we're taking into account, is like, how can we switch off these mechanistic relationships, these synergistic relationships? But as is quite common, it's not just you, there's just not so much knowledge about snakebite out there. It's not something as heavily researched as cancer. So there's a lot of questions that remain open, and a lot of fundamental science that needs to be done to understand it properly. So I hope, does this answer your question?
Yes, of course. Yes.
Perfect.
We have
Thank you.
another question from Vasilis Kranidis. If you can also turn on your camera. Thank you.
Yes, hello. Very nice presentation. I would like to ask, can this be applied on foodborne pathogens or neurotoxins, like from Clostridium botulinum or something
Yes.
from the aquatic? Yes.
Yeah. You can throw it in any toxins. You can, I mean, the thing is, you need to think about where is your target. So some of the toxins that you might be thinking of actually act within the cell. If they are in a cell, you have a problem, because a protein won't just get into the cell. So you can make your mini binders that bind it, but they won't get to it unless you catch it before it enters the cell, which in some cases is possible and some others not. But in theory, if you have a protein target, and that can be anything as small as a very small neurotoxin, like 5 kilodalton big, or as big as hundreds of kilodalton if you want. As long as you can get to it, then you can design things that bind to it. And it obviously helps if you understand the mechanism of action, because, for instance, some of these toxins target receptors. If you bind your mini binder, as they're called, or your design binder at the back, it won't do anything, right? Like it still can bind the receptor. But if you block it from actually binding to the receptor by going into the same region, then you're in business, because then it can't do anything. So as long as you keep these questions in your head when you're designing, yes, you can technically design anything against anything. Just make sure that it works in the system that you're actually targeting.
Thank you. Yes.
We have a question in the chat box. You can read it, I guess, Timothy. Just to all, if you have additional questions and the time is not permitting it to just discuss them, is this okay with you, Timothy, if they email you? You all have Timothy's email. You can email him gently and with good manner directly, right?
Yeah, no, not a problem. You're more than welcome to reach out. And I see the question in the chat is the name of the tool. So it's called RF diffusion. And if you just look at the Baker lab, you'll quite quickly see some of their publications. And you can also just look at RF diffusion. I can also type it into the chat so you can see it. But if you just look at that in GitHub, you will be able to find it quite quickly. Here you go. So hopefully that answers it.
I do have some questions. I will put them all together to you. There are two general questions. What it takes to make a research idea a company idea, or an alive and growing company. And because we don't always have the opportunity to talk with very big profile PIs, I was told by my boss in Finland, the most difficult thing, Spiros, is to hire the right people for your group. So can you tell us a few words about your hiring strategy? What do you see to your collaborators, to your PhD students, when some of us or someone else is sending an email to you? What do you look at the interview? And fast forward, my professor was looking that if the person who reached out to him by email from India, Pakistan, or Greece, or wherever, has read his research, his lab page, he comes with a problem, but also has gone to the length of providing a hypothesis, a funding source, and explanation. If you do those 5 things to his email, 99 times out of 100, you get an invitation for a Zoom interview.
Yeah, no, I mean, these are both really good questions. So I'll take them one by one. I'll start with your first one. How do you translate a good academic idea into an entrepreneurial one? It's a tough one, but I think the most important thing is to start thinking, okay, what is the problem? You've probably already figured that out, since that's already your academic question. But then thinking about your solution and really scrutinizing, not just will this work in the lab, but will this work in a real setting? Just like the question, the really good question I got before, can you use this for other toxins? Yes, yeah, you can. But think about the actual way that it's going to be administered. Is it going to be able to get to where you need it to get? It's really thinking these several steps ahead versus just doing it iteratively and looking, okay, what's the next step? What's the next step? That doesn't work. You really need to have the end game in mind. Also, then you might have something that works perfectly, but if it's, as I mentioned, not cost effective, or there's something else out there that you're not a lot better than, and nobody's going to do it. You need to really have something that's disruptive. So you need to think about, how is your solution going to change the way that we're doing something entirely? And if it's just marginal, there's a good chance, even if you're like 5% better, if those 5% don't mean much in terms of economy, nobody's going to care. If they mean a lot in terms of economy, people will care. And that's the sad truth, really, that money dictates a lot of these questions. So how are you going to get profit out of your idea? That's a question that you need to answer to investors. The nice thing is that it's just a different way of arguing the same problem. With snake bites, I don't care about financial gain. I don't think anybody who is into solving snake bites should care about financial gain. The idea is that we make it cost effective. So we can generate enough money out of the product so it can sustain itself, maybe have some innovation in it, but actually gets to the people. So I think the real important thing is early on, think about what is the final stage? How will this go? Is it going to go into patients? Is it going to go, are you selling something to a hospital? What are the main problems right now? Are your ways of solving it addressing these problems? Are they changing the way that we need to look at these problems entirely? And also things like regulatory approval, right? So for instance, these mini binders here that I mentioned, the AI generated protein, they're awesome. I love it. And it could really speed up our work, because if we can suddenly start clicking buttons and get a lot of binders very quickly, that's brilliant. But they aren't known antibody scaffolds. So what will the FDA say when we come? Is this going to make our clinical trials a lot more complicated and a lot longer? Are we maybe saving a year or two in academic research now, but then losing 5 years in clinical trials? Then it's not worth it. So, and these questions sometimes are incredibly hard to answer, and you just have to take a guess, or ideally ask some people who might have an answer to you. So I think in general, reaching out to people, also like the speaker mentioned before me, like using your network, finding people who know things about things you don't know so much about, is always a good idea, just to get a sanity check and some other ideas and thoughts on it. And just the last point on it is discuss your ideas earlier on. Like that's why we do a mention club. Don't sit on your ideas. You might be working an idea that's totally useless for 5 years' time, but you've never told anybody who can tell you that. Or you might be sitting on the next unicorn company but you're too scared, or you haven't had the opportunity to discuss it with somebody, and then maybe somebody else has done it by the time you actually get out of your shell and talk about it. So really be proactive about discussing your ideas, getting feedback early on. Yeah, I know this wasn't the most structured answer, but I think it hopefully covered some of the most important points. And the second question you had was related to how do you hire? And that, I fully agree, is an art form. And I will obviously say that so far I'm very happy with the outcome. I've got brilliant students. I've got really, really cool people that I'm working with. But it is a challenge. It's really hard because you also want to provide equal opportunities. You need to minimize bias. So I think in terms of, there's two key aspects. What do you look for and how do you do this? So what do you look for is people who are obviously technically skilled, who have some technical background that is relevant, but as you mentioned, that actually care about your science. If you get an email, dear professor, I would like to do a PhD in biotechnology. Can you help me? Then my answer will be no, because you obviously don't care about what I'm doing and what I can, or what my research is about. You just care about just getting a PhD in this field. So show me that you've, as you said, maybe at least looked at the website, maybe looked at some of the papers, at least read the abstracts, and have an idea on why you find it exciting. So having technical skills, but also having passion, some creative ideas, and okay, this is what I would like to do, or you're doing this and I find this really inspiring and I want to go more into this area because my dream is. So it's really understanding the human connection to the technical skill that for me is very important. I need people who are passionate about their work, because that is the environment I want to work within. I want people who enjoy it and not just do it well. And then how do you execute on this is a more difficult way. Like, how do you screen for this? One, obviously, sometimes people make it very easy for you and it's very easy to tell. But I also have started standardizing the way I interview more and more and more to make it a lot more fair, a lot more equal, to minimize the risk of bias. And I think that's really important, that you have kind of a set of questions that you ask the candidates that come when you actually get to an interview stage or so. And you give them a specific amount of time and space, and you have that equal across candidates. Because if you make it very personalized, some people will react differently, shine differently. And that's not really a fair way to try and assess this. So that's my 5 cents on this. I don't know if it's helpful.
No, it's very helpful. I don't want to take more of your precious time. Thank you again. I hope I have the opportunity to reach out to you again, so we can perhaps develop a closer relationship on my institution with yours. I know firsthand by teaching some of the students, we have great potential here that could be of help to you, but also you're doing excellent things that we'd like to learn more about. So,
of course, no, I'd love to interact more. And any of the students who participated here today and you're looking to look for internships or thinking about doing your master thesis elsewhere, so please get in touch, even if it's not directly related with me. Maybe we can find some solutions. But as discussed, be specific. I think another important thing is timeframes. When do you want to do stuff? Send your CV along so people can have a look at what your background actually is. All of these things help a lot in helping you. So don't feel, I'm always happy to chat with you guys and see if we can grow the collaboration. And thank you so much also for having me. I really enjoyed this.
thank you again, Timothy.
Have a good afternoon.
Have a nice, good rest. Bye-bye. Okay, thank you all for participating and staying that late. I will email you the recording